Hello and welcome to Spotlight on Antibodies, a podcast from Biointron, where we bring you the latest antibody industry trends. I'm your host, Jovina Celestio, and this month in February, top news includes novel antibodies to treat obesity, hereditary angioedema, B cells, and inhaled antibodies. Recently, Helicor Biopharma emerged from stealth with $65 million in Series A funding. They are focused on the discovery and development of first-in-class glucose-dependent insulinotropic peptide GIP antagonists, with their most advanced asset, HCR188, being a clinical-stage monoclonal antibody designed to bind GIP. The company's portfolio includes GIP antibody conjugates, including GLP-1 and other combinations, designed to address specific obesity subpopulations. Individuals carrying genetic loss-of-function mutations in either GIP or GIPR are protected from obesity and exhibit lean phenotypes thought to arise from diminished GIP or GIPR-mediated triglyceride uptake in adipocytes. By antagonizing GIP in the periphery and blocking transit to the central nervous system, Helicor's compounds may uniquely prevent GIP signaling in the brain, potentially restoring leptin sensitivity that may support increased satiety as well as reduced consumption and fat deposition. Meanwhile, iBio Inc., an AI-driven innovator of precision antibody immunotherapies, in collaboration with AstralBio, announced the development of an antibody that inhibits the function of activin E, a promising therapeutic target for cardiometabolic disorders and obesity. Individuals with a protective loss-of-function mutation in the INHBE gene have a healthier cardiometabolic profile, including less abdominal fat, lower triglycerides, and lower risk of type 2 diabetes and cardiovascular disease. While targeting activin E poses technical challenges for antibody discovery due to its difficulty to produce outside the human body, iBio has identified 5 critical epitope regions on the activin E protein by developing synthetic epitopes replicating these regions. Another target for obesity is activin receptor 2A. Leica has a clinical collaboration with Eli Lilly and Company to accelerate the development of LAE-102, a novel monoclonal antibody as a novel treatment for obesity. Activin receptor 2A is a receptor that has been shown to play a critical role in muscle regeneration and lipid metabolism. LAE-102 has been shown to both increase lean mass and decrease fat mass in preclinical models. If successful, this could, in combination with a GLP-1R agonist, further reduce fat mass and significantly regain the lean mass loss induced by GLP-1R agonist. This positions LAE-102 as a promising complementary drug candidate for achieving quality weight control. A few months ago, Convo Therapeutics also raised $60 million in Series B financing to focus on agonistic GPCR antibodies. The proceeds are being used to advance 2 wholly owned programs through Phase 1 and 2 additional programs to IND approval, including molecules targeting GPR75 for obesity. G-protein-coupled receptors are the largest and most diverse group of membrane receptors in the body. They play a key role in a broad range of biological processes by binding to a wide variety of signaling molecules, yet they share a common architecture. GPCRs are comprised of 7 transmembrane helices that span the cell membrane and intervening loops that are presented towards either the extracellular or intracellular environments. Agonists bind to GPCRs at an EC-facing binding pocket and initiate conformational changes that are propagated to the IC loops. This subsequently triggers downstream signaling via G proteins or G protein-independent pathways. These novel antibody therapeutics are an exciting advancements towards the treatment of obesity. According to the Centers for Disease Control and Prevention, about 2 in 5 adults in the United States have obesity. Being overweight and obesity can cause long-lasting inflammation and higher than normal levels of insulin insulin-like growth factor, and sex hormones. These changes may lead to cancer. On January 24, 2025, the novel monoclonal antibody Andembry, or garadacimab, was approved in Australia for the first time. This drug is now approved for routine prevention of recurrent hereditary angioedema attacks in patients aged 12 and older with C1 esterase inhibitor deficiency or dysfunction. Andembry is a novel factor X2A inhibitory monoclonal antibody developed by CSL. It works by inhibiting the plasma protein factor, which, when activated, initiates the cascade of events leading to oedema at various sites of the body. Hereditary angioedema is a form of bradykinin-mediated angioedema. It is a rare, debilitating, and potentially life-threatening genetic disorder affecting approximately 1 in 50,000 people globally, caused by deficient or dysfunctional C1-INH, a protein in the blood that helps to control inflammation. Inadequate amounts of properly functioning C1-INH can lead to the accumulation of fluid in body tissues, causing considerable swelling referred to as angioedema. HAE attacks can affect many parts of the body and can spread to multiple sites, including the face, abdomen, larynx, and extremities. Patients who have abdominal attacks of HAE can experience extreme pain, diarrhea, nausea, and vomiting caused by swelling of the intestinal wall. These attacks that involve the face or throat can result in airway closure, asphyxiation, and, if left untreated, death. A recent study looked at the real-world effectiveness of lanadelumab, a subcutaneously administered human IgG1 recombinant monoclonal antibody that potently inhibits active plasma kallikrein. Kallikrein is a protease that functions to cleave kininogen, subsequently creating kininogen and bradykinin, a potent vasodilator. Uncontrolled plasma kallikrein leads to excessive bradykinin production. Takeda's lanadelumab, sold under the brand name Takhzyro, was previously approved by the FDA in August 2018 for the treatment of type 1 or type 2 HAE. This real-world study demonstrates that lanadelumab long-term prophylaxis is effective in improving AFR in patients with type 1 or 2 HAE on every 2-week dosing and dose interval increases. A third novel monoclonal antibody that was recently given FDA orphan drug designation is Astria Therapeutics' navenibart, or STAR-0215. Like lanadelumab, navenibart targets plasma kallikrein with the goal to provide rapid and sustained HAE attack prevention. In a healthy blood vessel, C1 inhibitor blocks the function of plasma kallikrein. However, in HAE, a missing C1 inhibitor allows plasma kallikrein to process HMWK, generating cleaved HMWK and releasing bradykinin. Bradykinin then binds to receptors, allowing fluid to leak through blood vessel walls and causing pain. Navenibart inhibits and blocks plasma kallikrein even in the absence of C1 inhibitor. Based on positive results from the AlphaStar Phase 1b and 2 trial in people living with HAE, navenibart is advancing to Phase 3 development, with trial initiation expected in 2025 and top-line results expected in early 2027. B cells are central players in both humoral and cellular immunity, traditionally recognized for their role in antibody production. However, recent research highlights their direct involvement at sites of inflammation, where they establish tissue-resident memory populations and contribute to immune responses in infection, inflammatory diseases, and cancer. The spatial organization of B cells within tissues significantly influences their function, disease progression, and therapeutic outcomes. A few weeks ago, researchers from Oregon Health and Science University published a paper describing how B cells do more than simply produce antibodies. They also play a role in peripheral tissues in homeostasis and disease, and the researchers identified cellular and molecular signals that are involved in regulating their activity. Multi-omic analyses and in vivo fate mapping reveal that B cells infiltrate non-lymphoid tissues and tumors, undergoing in situ differentiation into memory B cells and plasma cells that shape local immune responses through cytokine and antibody production. These cells also act as tissue sentinels, prepared for future immune challenges. Their function is influenced by interactions within lymph node-like structures, such as tertiary lymphoid structures, which regulate their role in disease. However, key aspects of tissue B-cell biology remain unclear, including reliable markers for identifying BRM cells and the molecular signals that control their maintenance. Understanding TLS maturation and BRM cell differentiation could advance immunotherapy, enhancing protection against infections and strengthening B-cell-driven anti-cancer immunity. Recognizing the localized nature of B-cell function adds critical depth to our understanding of their immunological impact. Meanwhile, researchers from the University of Southern California have found how B cells can be reprogrammed through genome editing to produce custom heavy chain-only antibodies beyond what is achievable through immunization. This approach allows modifications to both the antigen binding and Fc domains, expanding the potential for engineered antibody designs. Using HIV envelope protein as a model, they demonstrated that edited B cells maintain regulated B cell receptor expression and antigen responsiveness in a tonsil organoid model. However, dual expression of endogenous and engineered antibodies raised safety concerns, including potential competition and self-reactivity. While naturally occurring dual BCR B cells exist, their role in autoimmunity remains unclear. This study presents a simplified genome editing strategy for engineering B cells with flexible antibody designs, paving the way for novel therapeutic applications while necessitating further investigation into safety and efficacy. Another recent study, led by researchers from Northwestern University, explored the potential of B-cell-based therapy that produces antibodies to inhibit glioblastomal growth. BVAX, a B-cell-based vaccine, effectively migrates to GBM tumors, differentiates into antibody-secreting plasmablasts, and produces antibodies that target extracellular matrix components critical for tumor invasion and motility. These antibodies inhibit GBM cell migration and invasion, demonstrating therapeutic potential. BVAX-derived antibodies also recognize both extracellular and cytoplasmic antigens, though the mechanism behind this remains unclear. Beyond antibody responses, BVAX may also enhance CD8 plus T-cell activation, contributing to its anti-tumor effects. Despite variability in antigenic reactivity among patients, BVAX consistently targets key regulators like fibrinogen and collagens. Future research should focus on optimizing BVAX's efficacy in GBM's immune-suppressive environment and identifying additional tumor antigens for therapeutic development. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow Biointron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.